All right, so let's move on with some more historical explanations. So Hippocrates was in 450 BC. Let's come a little bit more recently and talk a little bit about Immanuel Kant. So he was doing his philosophizing in the 18th century, so the 1700s. And again, he argued for four basic personality types. And in fact, some people say that he's the one who actually named the four personality types that Hippocrates described. So, he's the one who offered sanguine, choleric, melancholy, and phlegmatic. So some people say he's actually the one who named them. And he was accepting this idea of the four basic personality types. Let's come a little bit even more recently, and actually somebody who's associated with psychology, and that would be Wilhelm Vunt. He is credited by a lot of people for being the founder of psychology as we know it, because he attempted to study things in sort of a scientific way. He was doing his research in the 19th century, so 100 years, maybe even more from Immanuel Kant. He set up his lab in 1879. So, he was a very early psychologist. He thought there were two dimensions of personality. So there's the speed of change of the emotions, and then the strength of the emotions, that are the two personality dimensions. If you imagine those two dimensions being, double-headed arrows, and they intersect perpendicularly with each other, you end up with four quadrants created by those two dimensions. And you end up again with four basic personality types. So you have some people who have, fast changes of their emotion and strong emotions. You have ones who have fast change but weak emotions. You have those who have slow changes to their emotions, but they're strong emotions when they have them. And then you have those who are slow to change and also have weaker emotions. So you have like these four personality types that emerge again, based on his idea that there's two dimensions to personality. Even more recent, mid 1900s to, I think he might even still be alive today, Hans Eyck. I'm not positive. Probably should have Googled that before I started this lecture. But he, again, offered that there's two dimensions to personality, but he's a little different from Wundt in the sense that he labeled one dimension the introversion-extroversion dimension, and the other one the neuroticism or emotional stability dimension. So you end up with four basic personality types. Again, you end up with like the introverted, emotionally stable, the introverted, emotionally unstable, the extroverted, emotionally stable, and the extroverted, emotionally unstable. But these are in both Wundt and Eyck. These are, you should not think about the dimensions being only the polar ends. You should think about them being more like continuums, where you could be placed anywhere along that line. So you could be anywhere along that dimension of introversion and extroversion. You don't have to be a complete introvert. You could be maybe leaning towards introversion or leaning towards extroversion, and so on. So it's a lot more nuanced than just four strict personality types, but that's a good thing to keep in your mind for now. Buss and Plomin in 1984 offered the idea that there are individual differences in emotional intensity in the first days of life that are observable. And that we can tell in the first few days of life which children are wired up to have more emotionality, more activity, and then ultimately more sociability. So that's a pretty brave claim, that you can probably measure future emotional intensity in a very, very newborn baby. So I thought I had something else linked here, sorry. So Buss and Plomin thought it's so biological, that these traits that are listed here are so biological that they are present at birth, that they don't take any kind of influence from the environment to develop. All right, which brings up the issue of, okay, if it's biological, how much of individual differences can be attributed to genetic differences? That's what we mean by the word heritability. Heritability is an assessment of how much of individual differences can be attributed to genetic differences. So, one of our best sources of information about the heritability of traits would be twins. If you are a twin, shout out to you right now. Most twins that I've met have been dizygotic twins. That means that they were two separate eggs that each got fertilized by separate sperm. So they share about 50% of their genes. They're very similar that way genetically to regular siblings. The big difference between dizygotic twins and regular siblings is that they were raised, they spent time in the womb together, and they are the same age, and they're referred to as the twins. And so their environments are a lot more similar than regular siblings are probably going to be, even when they are mixed sex pairs, which is just as common in dizygotic twins as same-sex pairs are. Even when they're mixed sex pairs, the environment still tends to kind of treat them like a pair, right? Like not like two separate individuals. Monozygotic twins are created when you have one egg that gets fertilized by one sperm, and then at some point during development, it splits into two individuals. And when I say at some point during development, I mean within the first 14 days after conception, the split has to happen. They share 100% of their genes in common. They are, and I'm stammering a little bit because it's not perfect. We say 100%. If you are a monozygotic twin, you might be aware that, you don't share 100% of your genes with your twin. It's only 99.5 or something like that. But we're just going to round it up and call it good enough. So when you have dizygotic twins who are reared together, they are sharing the same environment from birth on, right? Like they're in the same household being raised together. They're going to allow us to look at the influence of environment on differences. So we can kind of look at dizygotic twins who are reared together versus dizygotic twins who are raised separately. Sometimes when babies are given up for adoption and it's a twin set, the babies will be given to different families to raise. And so we can kind of look to see whether those dizygotic twins are more or less different than the ones who are raised together. And that gives us a little bit of input on the environment. Monozygotic twins tend to give us information about genes. If they are reared together, their environment's overlapping. But if we look at monozygotic twins and we see that they were raised apart and they are more similar to each other than dizygotic twins who were raised together, we're going to say, well, gosh, the dizygotic twins, they shared environment even, and they're less similar to each other than the monozygotic twins. I'm going to say that the characteristic that we're measuring must be largely influenced by genes, right? So that's the kind of question that that researchers have posed with regard to twins. Are monozygotic twins who are raised apart more similar than dizygotic twins who are raised together? Now, I pulled a picture that just happened to have the dizygotic twins depicted as one male, one female. For the purposes of research, because it would be a confound to have the dizygotic twins including mixed sex pairs when that's impossible for a monozygotic twin pair. They have to be the same sex because sex is determined by genes. They make sure that in the dizygotic twin pairs, they only look at same-sex dizygotic twins. So that makes it less confounding. Well, what I wanted to share with you was the results of some research on this. And so what we're looking at in the red bars would be identical twin pairs. In the blue bars are what is called fraternal twins. Oftentimes that's the dizygotic twins. So the DZ twins are the fraternals. The MZ twins are the identicals. And along the x-axis, you see these correlations. So what they've done is they've taken the score for each twin and correlated it with each other on whatever measure: height, adjusted weight, intelligence, and so on. So they're correlating their scores with each other. Numbers that are closer to 0 are less related to each other. Numbers that are closer to 1 are more strongly related to each other. So first thing you can do is just go through and looking at each red and blue bar pairing. You can see that the red bar is longer in every case, right? So that means that there's higher correlations between the identical twins than there are between the fraternal twins. And by the way, just as a reminder, the identical twins in this study were raised in separate households, and the fraternal twins in the study were raised together. So even though they were raised in different environments, the identical twins are more similar. Now, you might not be surprised that the height matches so well for identical twin pairs because they have, almost 100% of their genes in common. In fact, you might say, wait, how is it not a perfect one correlation? Height is somewhat influenced by the environment, right? If you're sick at a critical moment in development, in growth, my son has a pair of identical twin brothers who are really good friends with him. One of them had an injury from Taekwondo that caused him to not be able to, run and play and do stuff for like a good solid 6 weeks. It might have even been longer than that. And in that time, his brother gained on him about half an inch. And it must have, it was like a critical period for them, as far as like they must have been in a really big growth spurt, because the uninjured brother grew taller during that period of time. And for the rest of their lives, he's now, well, not the rest of their lives because they're only like 26, hopefully lots of years ahead of them. But they are, to this day, I'll say, the uninjured one is about half an inch taller than the injured one. So it's not perfect. There is some environmental factors that go along with that. Same thing, even more profoundly, is adjusted weight. That weight is much more influenced by the environment. But look at intelligence. It's almost as closely correlated for the identical twins as height is. So it really underscores that idea that intelligence might be biologically based. Extroversion is, 0.5 correlation. Extroversion is a measure of how outgoing you are. Extroversion is more closely correlated for identical twins than schizophrenia is. And if you're a psychology student, you probably believe that schizophrenia is almost entirely biological. And that's what most of us think. And it's less correlated than extroversion is. Actually, it's less correlated than depression is. And less correlated than happiness is. So there are a lot of factors that are correlated for identical twins in a really strong way. But I want to make sure that you're aware how exactly to interpret these correlations. What we really want to do is take that correlation coefficient and then square it. And that'll tell us how much variability is accounted for by genes. So, for example, just pulling happiness, because it's almost 5, it's almost 0.5. Depression is 0.5. So if we square that correlation coefficient there, we will end up with 25, which means 25% of happiness, 25% of depression is attributable to genes. 25% of extroversion is attributable to genes. If we go to height and we square that 9, we're going to find that about 81% of the variability is due to genes alone. 81%, right? Like that's pretty good. We're getting up there when we do height. When we're doing, extroversion or whatever, 25% of it is attributable to genes. And then we have to start going, well, that sounds like there's got to be something else having an impact. Intelligence is more than 8. I'm just going to round it to 8 because it's easier for my math. So about 64% of intelligence is attributable to genes. So that's a pretty good amount of variance or difference between people that's being attributable to genes as far as that trait called intelligence. All right. Let's go ahead and, well, you know what? Let me do this really quickly because I'm not going to go through every teratogen that's possible here. But what I wanted to mention as far as like environment is don't forget that it starts in the womb. So when you have identical twins and fraternal twins, they're in the womb at the same time, right? And so things that happen while in the womb would presumably affect both of them. This gets a little bit esoteric that dizygotic twins are always in their own amniotic sac and they always have their own placenta, which means that while they're in the womb at the same time, their experience is not exactly the same. One placenta might filter out certain things that the other placenta doesn't. So dizygotic twins are going to have a more independent experience in the womb, as much as that can happen. Monozygotic twins can go all the way from being just like dizygotic twins, being, on their own placenta in their own amniotic sac, all the way up to being on the same placenta and in the same amniotic sac. So monozygotic twins can have a lot more variability as far as how much they share exactly the same uterine environment. So when I say things can be filtered out, I'm really referring to teratogens that can be harmful. Teratogens are substances that interfere with normal development. So they can be toxins, they can be infections, they can be radiation. Toxins include things like alcohol and other kinds of drugs. Fertilizer exposure can have teratogenic effects. What I really want to point your attention to is, first off, you'll notice that the effect on the body tends to happen in the first few weeks of life. So we're talking about the heart, the arms, the legs, the lips, the ears, eyes, teeth, palate, sex organs. All of this stuff is really basically going to be affected by teratogens. By the end of the 9th week, really the body's in place. But what happens throughout development for the entire gestational period, all the way up until birth, is the developing brain. And one of the things we think may happen in that last part of pregnancy, the last trimester of pregnancy, is that exposure to teratogens may have impacts on learning capabilities and then also personality factors. And so when we talk about biology and we say that these twins are exactly the same, we have got difficulty pulling apart things that might have happened in the womb that nobody could observe or know about necessarily, that might have impacted the twins differently. So when we see a lack of, perfect correlation between the identical twins on something like intelligence, it might be in part because they had exposure to different teratogens, for example. When we see a lot of variability on extroversion, again, it could be, they had different experiences in the womb, even though they were in there at the same time. So, all right, that's my last thing I wanted to say about twins. So let's go ahead and stop here. We'll come back and we'll talk about another angle on studying heritability, which is adopted children.